Hello, everybody. We're going to talk about the technique for gene transfer. And simply put, it's just transferring a gene from one species into another. Why would we do this? Many reasons. Creating genetically modified organisms. Oh, wow. Creating genetically modified organisms are also for medicinal purposes. And we're going to use the example of insulin. So if you remember, the central dogma for genetics is that gene equals protein. And gene is in your DNA. However, in eukaryotes, that DNA is trapped in your nucleus. So if you remember that we need to make a piece of messenger RNA, way back when, that messenger RNA will travel to a ribosome, eventually make a chain of amino acids, and will make our protein. So that's why we say gene equals protein. It's because of those steps. So the reason we can do this is that the genetic code is universal. Let's talk about what that means first. So let's talk about the amino acid valine, for example. So we're making an amino acid, and we're making a protein, and one of the amino acids in it is valine. Right? Protein is made up of all these different types of amino acids. But if one of these is valine, we need a piece of messenger RNA with the codon GUU. When we read these, we start from the middle and we work our way out. So GUU will make the amino acid valine. Also, so will GUC, GUA, and GUG. So there's lots of combinations that can make it. So then we can go like this. And the piece of DNA that will eventually have the information to make that messenger RNA, complementary base pairs still work for this, would be cytosine, right? Complement to guanine, adenine complements to uracil in RNA, and another one. So what this is saying is that this code, this genetic code, this piece of DNA will make valine in every living species on this planet, with the exception of some weird bacteria at the bottom of the ocean. So that's the how. Like, we can do this because the genetic code is universal. Let's look at an example with insulin. So if we look at the assessment statement up here, it says we need to include some stuff. So we need to talk about the role of plasmids. We need to talk about two different types of enzymes, so plasmids. We need to talk about the host cell. We talk about bacteria, two enzymes called restriction enzyme, and also DNA ligase. So what we need to do is we need to take a plasmid, like you remember, plasmids are in bacteria, and we need to remove it. Something missing from this picture is that we need to take the gene for human insulin, because that's what we're going to transfer, out of the human genome, out of one of the chromosomes, and we need to eventually put it in this plasmid. But in order to put it in there, we need to cut a hole right in this plasmid. If you see those little scissors, we've created like a gap in our circle. Also, we need the same little scissors, and we need to cut out our gene. So here's the gene for insulin in this example. Oops. And we've cut it out using these scissors. These scissors are your restriction enzymes. That should ring a bell from gel electrophoresis, right? When we're cutting DNA in different lengths, we use the same technology. So we've cut two spaces, and now we've just glued, or at least inserted, the insulin gene into this plasmid. Now, one of the things we also need to do is that we need to glue it in here, like the sides. So this is all bacterial DNA, the yellow. The red is our human insulin. We need to glue it in, and to do that, we need the other enzyme called ligase. Ligases are glue. Then we simply, well, I can say simply because I don't do the technology, smart scientists do. We take this plasmid and put it back into a bacterium. And if you can see, there it is, right? The recombinant, that's the word we use for DNA that's been changed, the recombinant DNA with the bacterial DNA, but also human, it's in this bacteria, and the bacteria divides through binary fission. And we make lots and lots and lots of bacteria, all with the same information in it to make human insulin. And just like magic, Bacteria make human insulin.